the manifestation of life. All organisms, the simplest as well as the most intricate ones, are slowly changing all the time they are alive. Unlike many animals, plants typically continue to grow throughout their lives by absorbing water and minerals. From ingredients such as water, minerals, proteins, carbohydrates, fats, vitamins, hormones, etc., organisms form additional protoplasm. The formation of protoplasm is called assimilation. A large part of the food which a plant manufactures is used as a source of energy by the plant and other living organisms. This surplus food may be built more or less permanently into its tissues, producing new protoplasm and new cell walls, and thus promoting the growth of the plant body. Growth represents excess of constructive metabolism over destructive metabolism.
Dear students, I welcome you all to study about plant growth, characteristics of growth, phases of growth and measurement of growth. ਪਿਆਰੇ ਵਿਦਿਆਰਥੀਓ, ਅੱਜ ਦੇ ਵਿਸ਼ੇ ਵਿੱਚ ਅਸੀਂ ਗੱਲ ਕਰਾਂਗੇ ਕਿ ਪੌਦੇ ਕਿਸ ਤਰ੍ਹਾਂ ਵੱਡੇ ਹੁੰਦੇ ਹਨ, ਜਿਸ ਨੂੰ ਪੌਦੇ ਦਾ ਵਾਧਾ ਕਿਹਾ ਜਾਂਦਾ ਹੈ। ਜਦੋਂ ਪੌਦੇ ਦਾ ਵਾਧਾ ਹੁੰਦਾ ਹੈ ਤੇ ਇਸ ਦੇ ਨਾਲ ਪੌਦੇ ਦਾ ਵਿਕਾਸ ਵੀ ਹੁੰਦਾ ਹੈ। ਇਸ ਲਈ ਅਸੀਂ ਇਹ ਕਹਿ ਸਕਦੇ ਹਾਂ ਕਿ ਵਾਧਾ ਅਤੇ ਵਿਕਾਸ ਇੱਕ ਦੂਜੇ ਨਾਲ ਲਿੰਕਡ ਹਨ। ਇਸੇ ਵਿਸ਼ੇ ਤੇ ਵਿਚਾਰ ਕਰਨ ਤੋਂ ਪਹਿਲਾਂ ਆਓ ਅੱਜ ਦੇ ਪਾਠ ਦੇ ਸਿੱਖਿਆ ਉਦੇਸ਼ਾਂ ਤੇ ਨਜ਼ਰ ਮਾਰ ਲਈਏ।
Learning objectives. On completion of today's class, the learners will be able to define growth, discuss the characteristics of growth, explain the phases of growth, discuss the methods for measuring growth. Life starts from a fertilized egg or zygote, which through a series of precise and highly ordered divisions develops into a mature plant or animal. Growth is the fundamental property of all living cells, and all developmental processes involve growth. A tiny plant finally develops into a mature plant. Stages in the life of a plant whose body is very complex shows that it bears flowers, fruits, senescence, and eventually dies. Thus, three terms, that is, development, differentiation, and growth, are used to describe the various aspects of changes that plant undergoes during its life cycle.
How can we define growth? Growth is defined as irreversible increase in size by cell division and cell enlargement together with the synthesis of new cellular materials. Growth involves an irreversible increase in size, which is usually but not necessarily accompanied by an increase in dry weight. The basic factor which is responsible for growth is the production of new protoplasm, which is clearly evident in the regions of active cell division. The next stage in growth is increase in plant size, which is the result of absorption of water and the consequent stretching of tissue, a process which is actually not the growth at all. The third and last stage in growth involves the entry of plenty of building materials, chiefly carbohydrates, into the expanded young tissues. This results in an increase in the dry weight but no visible increase in external size of the plant.
Growth can be observed by a variety of quantitative measures. These are cell division. Growth takes place in plant by activity of meristematic cells. This activity continuously adds new cells to a growing tissue. Cell enlargement. Enlargement in organs occurs because of cell enlargement. Fresh weight. Sometimes growth is measured in terms of fresh weight, especially in cell culture. Dry weight. Growth is mainly defined as irreversible increase in dry weight. increase in length or width, it can be observed in case of seedling growth.
ਪਿਆਰੇ ਵਿਦਿਆਰਥੀਓ, ਪੌਦਿਆਂ ਵਿੱਚ ਵਾਧੇ ਬਾਰੇ ਹੋਰ ਚੰਗੀ ਤਰ੍ਹਾਂ ਜਾਣਨ ਲਈ ਸਾਨੂੰ ਵਾਧੇ ਦੇ ਸਾਰੇ ਫੇਜ਼ਸ ਨੂੰ ਸਮਝਣ ਦੀ ਲੋੜ ਹੈ। ਹੁਣ ਅਸੀਂ ਗੱਲ ਕਰਾਂਗੇ different phases of growth ਦੀ।
The plant is a community of different cell types formed by cell division and subsequent differentiation. The actively dividing or meristematic cells localized in regions called meristems retain their ability to divide repeatedly and give rise to new cells throughout the life of a plant. The cells of meristematic tissue, which lie at the apex of the roots and shoots, are known as apical meristem. Some meristematic cells are also present at the base of leaves or stem and in the internodes of some monocotyledonous plants. Thus, dicot stem grows from the tip, while monocots may grow either from the tips or base of the stem or internode. Nodes are regions of the stem at which leaves are attached. These may also contain meristematic cells. Axillary buds are found in the axils between leaf and stem, and under appropriate conditions, these buds can be activated to produce branches. In the highly organized context of plant, each cell of root or shoot goes through an orderly series of development phases. The daughter cells derived from cell division in the meristems enlarge and bring about visible increase in size of the plant. The expanding cells differentiate to form the different tissues and become part of a mature organ.
Increase in girth of dicotyledonous stem and roots is produced by tangential division of meristematic cells in the cambium, a one-cell thick cylinder of tissue that lies between xylem of the wood and phloem of the bark. The cells produced by cambial divisions differentiate into either secondary xylem present inwards or secondary phloem present outwards. Let us explain what are meristematic cells. Meristematic cells are small, almost cubical in shape, thin-walled, multivacuolate. Their nucleus is large as compared to the rest of the cells. Now we will study about phases of growth. Growth can be studied in following three phases: cell division or formative phase, cell enlargement or phase of elongation, cell maturation or phase of differentiation. ਪਿਆਰੇ ਵਿਦਿਆਰਥੀਓ, ਹੁਣ ਅਸੀਂ ਪੜਾਂਗੇ ਕਿ ਸੈੱਲ ਡਿਵੀਜ਼ਨ ਫੇਜ਼, ਜਿਸ ਨੂੰ ਫਾਰਮੇਟਿਵ ਫੇਜ਼ ਵੀ ਕਿਹਾ ਜਾਂਦਾ ਹੈ, ਉਸ ਵਿੱਚ ਸੈੱਲ ਕਿਸ ਤਰ੍ਹਾਂ ਡਿਵਾਈਡ ਹੁੰਦੇ ਹਨ ਅਤੇ ਆਪਣਾ ਨੰਬਰ ਕਿਵੇਂ ਵਧਾਉਂਦੇ ਹਨ।
In cell division phase, new cells are formed by the repeated divisions in meristematic cells. A meristematic cell is a small isodiametric cell with thin and elastic cell wall, thick protoplasm, prominent nucleus, and minute but with numerous vacuoles. Metabolically, these are highly active cells. To produce new cells, they assimilate large quantities of carbohydrates, proteins, mineral salts, and water. They respire actively to meet the energy requirements for assimilatory activities. During mitotic cell division, the cellular mass of a meristematic cell increases and the replication of genetic material takes place. As a result of this division, each of the two daughter cells becomes only half the size of the parent cell. These cells then enlarge and synthesize their own cytoplasm and cell wall materials. Daughter cells thus produced may remain meristematic or may enter into the next phase of growth.
ਹੁਣ ਅਸੀਂ ਪੜਾਂਗੇ growth ਦੇ ਦੂਸਰੇ phase cell enlargement ਬਾਰੇ. Some of the daughter cells of meristematic region stop dividing and start increasing in their size. Enlargement is clearly visible in linear direction and hence the phase is known as phase of elongation. During this phase, the newly formed cells absorb water, which results in the increased turgidity. In the initial stages, the cell enlargement occurs in all directions, but in later stages, it is more prominent in linear direction. Cells under this phase show high rate of respiratory and assimilatory activities associated with growth regulators and enzymes.
Cell enlargement is driven by water uptake and limited by strength and rigidity of cell wall. Small vacuoles fuse to form a large vacuole, pushing the protoplasm to the periphery. Osmotic pressure of cell sap in the vacuole increases and consequently water enters in the cells. Not only the absorption of H2O but the synthesis of new cytoplasm and cell wall material also accompany the cell enlargement. Growth due to cell enlargement is very high.
ਹੁਣ ਅਸੀਂ growth ਦੇ third phase ਜਿਸ ਨੂੰ cell maturation ਕਿਹਾ ਜਾਂਦਾ ਹੈ ਬਾਰੇ ਗੱਲ ਕਰਾਂਗੇ। ਇਸ phase ਨੂੰ phase of differentiation ਵੀ ਕਿਹਾ ਜਾਂਦਾ ਹੈ।
Differentiation is the process by which cells that appear to be similar assume different morphological patterns and physiological roles. Differentiation is one of the most difficult puzzles in biology. A newly formed cell may develop along any of several morphological and physiological lines depending on genetical, physical, chemical, such as internal gradients of hormones and other metabolites and spatial influences around it. Once a cell has differentiated along a specific pathway, it usually does not revert to the undifferentiated state and assume a new form. Current thinking revolves about the theory that in all cells, only a portion of the total genetic potential is expressed, some genes being turned on and others off. The nature of a cell thus depends on the complex of genes that is active.
Differentiation usually starts along with the elongation in phase. During growth, cells undergo structural changes such as change in shape, size, and cell wall, and physiological, that is metabolic and biochemical differentiations. Structural differentiation usually accompanies with phase of elongation, as at the end of phase of elongation, the cells attain a definite shape and size to perform particular function. Depending upon the type of cells, wall grows in thickness along with the development of other characteristic features. Physiological differentiation is not restricted but continues in all phases of growth. During this phase, assimilatory activities become restricted to assimilate only stored food, that is carbohydrates and fats. Rate of respiration is also less than in the previous phase of growth. Dear students, now we will study the course of growth. ਜਿਸ ਤਰ੍ਹਾਂ ਤੁਸੀਂ ਜਾਣਦੇ ਹੋ ਕਿ ਪੌਦਿਆਂ ਵਿੱਚ ਵਾਧਾ ਅਲੱਗ-ਅਲੱਗ ਦਰ ਨਾਲ ਹੁੰਦਾ ਹੈ, ਜਾਂ ਅਸੀਂ ਇਹ ਵੀ ਕਹਿ ਸਕਦੇ ਹਾਂ ਕਿ ਇੱਕ ਹੀ ਪੌਦੇ ਦੇ ਅਲੱਗ-ਅਲੱਗ ਹਿੱਸਿਆਂ ਦੇ ਵਧਣ ਦੀ ਦਰ ਵੀ ਵੱਖ-ਵੱਖ ਹੁੰਦੀ ਹੈ. For example, vegetative growth of most plants in general shows three phases, starting slowly, becoming gradually faster, and finally slowing again. These three phases together are known as grand period of growth. It covers the whole of the vegetative history of an annual plant. In a perennial plant, grand period of growth is repeated annually with periods of dormancy between the repetitions. In 1873, Sash studied growth systematically and designated the total period of growth as grand period of growth.
SATs plotted different phases of growth against time and obtained a sigmoid curve or S-curve. It consists of lag period. In sigmoid curve, rate of growth at the initial step is very slow. This period is called lag period. It denotes the period of formative phase in which organ prepares itself for a rapid growth. Log period. In this period, rapid growth takes place. Maximum rate of growth is achieved during this period and it denotes the period of elongation phase. Steady period. Subsequently, the rate of growth declines and finally stops. This period is known as period of diminishing growth. During this period, differentiation is accomplished. Organs of limited growth do not show further growth and a steady state is reached. Whereas in the organs of unlimited growth, the period of diminishing growth continues till senescence.
Dear students, now we will discuss the different factors which greatly influence the plant growth. The plant growth is influenced by a variety of external and internal factors.
These are categorized as shown. Environmental factors such as light, temperature, water, oxygen supply. Nutritional factors such as carbon-nitrogen ratio, soil nutrients. Now we will discuss each environmental factor in detail one by one. Let us discuss factor affecting plant growth. Regardless of the habitat in which a plant is growing, variabilities of a complex set of environmental factors play an important role in the growth and development of any plant. Important among these environmental factors are temperature, light, oxygen, water, and nutrients.
Students, temperature is one of the most important factors that affects the growth of any plant. However, the minimum, optimum, and maximum limits of temperature for growth vary from species to species. A temperature of 28 degree to 30 degrees Celsius is optimum for proper growth in most plants. The rate of growth in plant tissues increases with increase in temperature to optimum values, after which it declines as the temperature becomes excessive.
Arctic and alpine species may grow at the freezing point or even at a temperature slightly below the freezing point. Their optimum temperature is usually no longer than 10 degrees Celsius. The optimum temperature for most of the tropical species varies from 30 degree to 35 degrees Celsius, and for temperate species it usually varies from 25 degree to 30 degrees Celsius. Effects of duration for which a plant is exposed to a particular temperature also vary with the species. Soil temperature also greatly influences the growth of roots and shoots. Fluctuations in day and night temperature favors better growth. This fluctuation of temperature influencing growth and metabolisms is termed as thermoperiodicity.
ਪਿਆਰੇ ਵਿਦਿਆਰਥੀਓ, ਹੁਣ ਅਸੀਂ ਪੜਾਂਗੇ ਕਿ ਲਾਈਟ ਕਿਸ ਤਰ੍ਹਾਂ ਗਰੋਥ ਨੂੰ ਇਨਫਲੂਐਂਸ ਕਰਦੀ ਹੈ. Light is another important factor that variously affects growth and development of all plants. Light intensity, quality of light, and duration of light affect growth and development of all plants. Light greatly influences several important physiological processes like chlorophyll synthesis, stomata movements, photosynthesis, formation of anthocyanin, temperature of aerial organs, absorption of minerals permeability, rate of transpiration, streaming of protoplasm, etc. Let us discuss all these factors.
Intensity of light. The intensity of light greatly influences plant growth. Variation in light intensity has more significant effects upon growth pattern of plants than changes in the quality of light. Most crops and ornamental plants, for instance, wheat, corn, peas, tobacco, show vigorous and stocky growth and flower profusely with full sun. Such plants are called sun plants. When grown with intermediate light intensities, sun plants become taller and have longer, thinner leaves but fewer flowers. They show very poor growth in low light intensity.
Students, next important factor is quality of light. Different wavelengths of sunlight have significant effects upon the growth of plants. Most of the experiments conducted in this direction indicate that overall development of a plant and increase in its dry weight take place. This increase in its dry weight takes place not effectively in the full spectrum of visible light. Plants grown in blue and violet tend to be dwarf. Those in red light grow tall. The ultraviolet and infrared radiations of sunlight do not promote growth. Overall growth of plant in green light is much less than in either blue, violet or orange-red portion of the spectrum. This effect of green light is tremendous due to lower efficiency of photosynthesis in the green light. Let us discuss next important factor, which is duration of light.
Duration of light. Duration, intensity and quality of light have marked influence on the rate of photosynthesis and hence the rate of growth. During winters, when the days are short, plants grow slowly. As the days get longer towards spring, growth is accelerated. Duration of light not only affects photosynthesis but also greatly influences dormancy and flowering in plants. The short days of autumn bring about retardation of growth in many plants. A number of trees respond to the short days of autumn by ceasing growth and becomes dormant. The length of day has a marked influence on flowering of plants. According to their requirement of light for flowering, plants are classified as long day plants, short day plants, day neutral plants. The long day plants in general flower when the days are longer than 13 to 14 hours. Short day plants produce flowers when the days are shorter than 13 to 14 hours. Flowering in the day neutral plants is not affected by the length of day. They can very well flower under both short and long day conditions.
Dear students, ਹੁਣ ਅਸੀਂ ਪੜਾਂਗੇ ਕਿ ਆਕਸੀਜਨ ਸਪਲਾਈ ਨਾਲ ਗਰੋਥ ਤੇ ਕਿਸ ਤਰ੍ਹਾਂ ਦਾ ਅਸਰ ਹੁੰਦਾ ਹੈ. Oxygen supply. With the exception of only those plants which are native to marshy and boggy terrain, the growth of all terrestrial plants is greatly retarded in poorly aerated soils. Usually, the shoots of plants receive an ample supply of oxygen, but the roots may or may not get sufficient oxygen to grow and function normally. Plants in flooded fields or in waterlogged pots do not thrive due to marked deficiencies in soil aeration. The related growth of plants in poorly aerated soils is chiefly due to reduced absorption of minerals and water. Oxygen is essential for aerobic respiration and hence availability of energy for biosynthetic activity. प्यारे बच्चों, ਅਸੀਂ growth ਤੇ ਅਸਰ ਪਾਉਣ ਵਾਲੇ ਫੈਕਟਰਸ ਪੜ੍ਹ ਰਹੇ ਹਾਂ. ਕੀ ਤੁਸੀਂ ਜਾਣਦੇ ਹੋ ਕਿ ਪਾਣੀ ਵੀ ਇੱਕ ਜ਼ਰੂਰੀ ਫੈਕਟਰ ਹੈ आओ इस बारे होर पढ़िए। Water is one of the most essential requirements for growth of a plant. Plants grow well when ample but not excessive moisture is available. Water is required for cell elongation, maintenance of turgidity of growing cells, and provide medium for enzyme action. Excessive transpiration or water deficiency in soil causes water stress in plants. Growth is retarded during water stress. ਹੁਣ ਅਸੀਂ ਡਿਸਕਸ ਕਰਾਂਗੇ ਮਿੱਟੀ ਵਿਚਲੇ ਨਿਊਟ੍ਰੀਐਂਟਸ ਦਾ ਪੌਦਿਆਂ ਦੀ ਗਰੋਥ ਨਾਲ ਸੰਬੰਧ।
Soil nutrients. The quantity and nature of soil nutrients have marked influence in the growth and development of plants. For luxuriant growth of any crop, field should be adequately rich in nutrients. For example, carbon and oxygen in CO2 and hydrogen in water are assimilated in photosynthesis. The ratio of carbohydrates and nitrogenous compounds regulate the specific pattern of growth in plants. If a plant contains more nitrogenous compounds as compared to carbohydrates, it produces more protoplasm, less mechanical tissue, and vigorous vegetative growth. On the other hand, less nitrogenous compounds and more carbohydrates favor synthesis of more wall material, less protoplasm, and more mechanical tissue. Growth depends upon the availability of essential nutrients. Nutrient deficient conditions retard the plant growth. ਇਹ ਜਾਣਨ ਲਈ ਕਿ ਪੌਦਿਆਂ ਵਿੱਚ ਕਿੰਨਾ ਵਾਧਾ ਹੁੰਦਾ ਹੈ, growth ਦੀ ਮਿਣਤੀ ਜ਼ਰੂਰੀ ਹੈ। Growth in plants can be measured in terms of increase in the length or girth, that is, increase in length of stem and root, increase in fresh or dry weight, increase in area or volume, that is, increase of fruits and leaves, increase in number of cells produced. Practically, any individual measurement is sufficient, but more information will be required to get correct result. Example, when a seed is germinated and allowed to grow in dark, it elongates rapidly. At the same time, the dry weight of the seedling decreases because it utilizes the reserve food stuff to grow.
The growth in a plant is usually measured in terms of increase in its length. Some of the most prevalent methods to measure growth are direct method and by oxenometer. In direct method, the length of plant or its part is directly measured with the help of a scale at a particular time interval to find out change in length. Oxenometers are mainly of two types. These are arc oxenometer and Pfeffer's oxenometer. But here we will discuss only one type, that is arc oxenometer. In arc oxenometer, there is a pointer fixed to a pulley round which passes a thread. The tip of the pointer moves freely on a graduated arc. One end of the thread is tied to the stem tip of a potted plant, and to the other end of the thread, a weight is suspended which keeps the thread tight. When the stem elongates, the thread goes down by the pressure of the weight, and consequently pulley and pointer move likewise. The pointer moves down on the graduated arc. The distance through which pointer moves is noted on the graduated arc. From the recorded readings on the scale, increase in the length of stem can be calculated. For example, suppose the scale shows an increase of 50 cm in 24 hours and the magnification is 100 times. Then the actual growth in 24 hours will be 50 divided by 100 cm, that is 0.5 cm. So students, this was about the plant growth, characteristics, phases of growth, measurement of growth, and this concludes our today's lesson. Let's summarize all which we have learned so far. Growth is defined as irreversible increase in size by cell division and cell enlargement together with the synthesis of new cellular materials. Growth can be observed by a variety of quantitative measures. Important ones are cell division, cell enlargement, fresh weight, dry weight, and increase in length or width. Meristematic cells are small, almost cubical in shape, thin-walled, multivacuolate. Their nucleus is large as compared to the rest of the cells. Growth can be studied in following three phases. Cell division, that is formative phase. Cell enlargement, that is phase of elongation. Cell maturation, that is phase of differentiation. Differentiation is the process by which cells that appear to be similar assume different morphological patterns and physiological roles. In 1873, Sash studied growth systematically and designated the total period of growth as grand period of growth. Different factors which influence growth are environmental factors and nutritional factors. The growth in a plant is usually measured by direct method and by oxenometer. Now we have reached to our destination, means end of today's topic. Here is a quick test for you to find out how much you have absorbed. Question 1. Name the fundamental property of all living cells. Answer. Growth is the fundamental property of all living cells. Next question is, define growth. Answer. It is defined as irreversible increase in size by cell division and cell enlargement together with the synthesis of new cellular materials. My next question is, what is the basic process of growth? The answer is, the basic process of growth is production of new protoplasm in the regions of active cell divisions. Next question, name three phases of growth. The answer is cell division or formative phase, cell enlargement or phase of elongation, and cell maturation or phase of differentiation. Next question, what are meristematic cells? Answer, meristematic cells are small, approximately cubical in shape, thin-walled and multivacuolate, and their nucleus is large relative to the rest of the cell. Next question is, what is grand period of growth? Answer, the total period of time during which all the phases occur is called as the grand period of growth. Next question, name three phases of S-shaped growth curves. The answer is lag phase, log phase, and steady phase. Next question, name the different factors influencing growth of plants. The answer is environmental factors and nutritional factors. Next question, name the types of oxenometers used to measure growth in length. The answer is arc oxenometer and Pfeffer's oxenometer. The next question is, how growth is measured in direct method. The answer is, the length of a plant or its any part is directly measured by means of a scale at a particular interval of time to find out change in length. Okay friends, with this I would like to wind up. See you in next class with another interesting topic. Thanks for your cooperation and attention. Goodbye.